Steve is a very interesting person to be with. He has a lot of enthusiasm for science. He has a breadth of knowledge, and he wants to share it with you. And he wants to learn what you are thinking about.
He's the perfect mentor. He always was a nice guy. He was a fair guy. There was no issue of women in the lab, and he's just a natural. He's a natural mentor, and he's a really good scientist.
He started out with a wonderful pedigree. He was a student with Paul Berg at Stanford, of course, and then a postdoc with David Baltimore at MIT. And so he could not have been better trained. It was always clear to me that this was a person who really understood, at a deep level, understood what the science was about and where it was going.
I was born in Providence, Rhode Island. I grew up in a small town in Massachusetts, Swansea, Massachusetts, on the water. My dad was a carpenter and a contractor, and we had an amazing shop. So we were always building things, repairing things, and always interested in how things worked. So I'd gotten into Harvard and others early, but I still held out in my mind that, gee, it would be interesting to go to a place like Stanford. I remember I had a phone call from Bob Lehman, who was doing the admissions at the time. And he said, we decided we're going to accept you. So take some time, think it over, and let us know as soon as you can whether you think you could come. And I said, that's okay. I'm coming. I'll tell you right now. I accept. Because at that point, I was sure I wanted to do it. The thread running through everything was enzymes. And that was Arthur Kornberg's love. He saw the power that enzymes had, the power they had to make molecules do things that you wanted them to do. I think he built an amazing department. He created a culture that was phenomenal. We learned how to manipulate the DNA of this virus, SV40. We learned how to cut it up into pieces. We learned how to create mutations in specific sites on the viral DNA. And then we could read out the consequences of what we'd done because we could transfect these DNAs back into cells in culture, recover virus, and see what we'd done to it, basically. So it was a way to learn the functions of each of the viral genes. And then the next step was to be able to actually insert foreign genes into the genome of the virus. And that led us into the thinking about building vectors for DNA that you could use to deliver into cells. When I was thinking about a postdoc, I made a long list of possible labs that I was interested in. And Baltimore's lab was near the top, certainly, and Paul looked at it at one point and said, yeah, David would be great. A month later, he came in and said, well, it's all set. I said, what? And he said, you're all set with David. And I said, okay.
I first met Steve in 1978 when he moved to MIT to be a postdoc in Dave Baltimore's lab. At that time, MIT was in the midst of some very exciting times. David had just received a Nobel Prize 2 years before. Steve is someone who's a very positive personality, always engaged, fun to talk to, and always in motion. His work here at MIT with David at the time identified the DNA sequence for the Abelson oncogene. And Abelson, the oncogene is fundamental when mutated in patients in creating CML, a very lethal leukemia. And one of the first breakthroughs in precision cancer treatments was the development of Gleevec, which inhibits that Abelson gene and is really very effective in controlling leukemia. That work is the signature work in advancing our treatment now of cancer. Steve's work has been foundational. It's a village at the top, but there are a few people who laid the foundation, and Steve was part of that, and then helped build the rest.
He's made some major discoveries, and particularly in how viruses and cells interact with one another. What's the machinery that works together? For a virus to get into a cell, make more of itself and get out of a cell, it requires that it fit in and do things that work with the cell. And by studying these processes in detail, you learn not only about the virus, but you can also learn very new insights in what the cell is doing. So you can discover new cell functions by studying how the virus is interacting in that. And he's done a lot of that.
He's found so many different host proteins and actually figured out how they work in terms of repression of transcription, silencing of the virus, integration sites. He's examined almost every gene possible in MLV, but he also did move on to HIV and continue to find proteins and figure out these different host proteins, how they work, and how the virus usurps them.
The Department of Biochemistry at Stanford had a huge impact on me, there's no doubt. You run your lab based on what you've seen very much. And Paul's lab ran very much the way I hope to run a lab now. It's a culture where you encourage your students to do what they want. You try to give them all the tools they need, the freedom to work on the projects they think are exciting, and good things happen.
I came to Steve Goff's lab, and on my first day, they had just did their first assay for reverse transcriptase that they had cloned. And I sat there with him, and then I started to list all the negative things that could go wrong. But Steve just said, okay. He just answered okay. And then I realized, oh my God, this is what I've been dreaming for. I've been dreaming for somebody who liked your ideas and gave you the breath to do it. And it wasn't like he wasn't critical, because he was critical. When you actually did the experiments, he was there with the questions, but he wasn't there suppressing your ideas.
I see the mentoring of students and postdocs as my primary job. The most exciting thing is always when you learn something new, something surprising. When a student or a postdoc brings something that's amazing and they're excited and you're excited, it's what you really live for.
I think Steve's a wonderful choice to receive this award out of the Department of Biochemistry and associated with Arthur Kornberg and Paul Berg, because he represents the sort of culture of that department in his focus on excellence, his focus on students and scholarship, and being innovative in creating science that is important unto itself, but also builds a foundation for other people to advance science.
It's really an honor to receive the Arthur Kornberg and Paul Berg Lifetime Achievement Award. I'm really honored. You go through your career after graduate school not knowing exactly how people thought of you back when you were a student. It's really a pleasure for me to feel that Paul maybe enjoyed my time with him, maybe appreciated the work we did together. And I think it was an important time for me. And I feel really proud, therefore, to receive this award. Thank you.